All right, we've already talked a little bit about the plasma membrane when we talked about the fats and lipids and things like that. So just as a reminder, the structure of the plasma membrane, it's described as a lipid bilayer composed of phospholipids. If you remember, the phospholipids had the fatty acid heads up here, and the nonpolar, the phospholipid, the phospho head up here, the fatty acid tail in here. This is nonpolar. This region is polar. This is hydrophilic. This is hydrophobic. Okay. And another thing that involves in that we didn't really mention is that there's proteins embedded throughout the material. And because of that, it's called the fluid mosaic model. So if you think about a mosaic, it's a bunch of little tiles all together to make a particular piece of artwork. When we talk about the plasma membrane, it's the same thing. There's a bunch of little pieces all together to form and get the whole thing, the whole process. So the proteins would actually kind of be embedded in this region. And I'll show you more in a different diagram. There you can actually see, so if you take a look at this, you can see this region right up here would be the phosphoheads. There's the tails in between, creating a bilayer that if you are hydrophilic, you'll get stuck in this region. If you're hydrophobic, you'll be stuck here, therefore acting as a barrier or a gate, and that's the purpose and function of the plasma membrane. Just another image there showing the fatty acid tails, phosphate head. All of this should be a review from a unit on fats. The phospholipids themselves, if you look at them, are made up of two distinct regions. You should be able to identify and label those regions on the test. There is a phosphate head up here that is also hydrophilic and is polar. Down here in this region, that's the tails, and that's going to refer to the down here in the tails, that is hydrophobic and it's non-polar. Okay, so those two different parts create the plasma membrane. The head region will be attracted to water inside the cell and outside. There's also cholesterol. Cholesterol is a non-polar. If you remember, we talked about it being like a lipid, and what it'll do is it'll actually embed inside the non-polar regions in here. So the cholesterol can be found in here, and it helps. A big part of it is it makes it strong and flexible. And I'll talk about kind of another thing that it does in regards to temperature in class. So remind me to, one thing you should write down is that to ask me about temperature, what role it has in temperature in class tomorrow. There you can actually see it, a great diagram. Make sure you have that in your notes that you can bring to class and show me the structure of a phospholipid with all the parts labeled. If you need to pause it now, I'm going to be moving on to the next slide. There are also membrane-bound proteins that are embedded in there. Remember, because of the different parts. You have some proteins that are on the surface that extend through to the extracellular matrix. And once again, their role is to play a major portion in the, they play a major role in the actual regulation and interactions between other materials. And so, for example, a membrane-bound protein on the surface of your plasma membrane may play a major role in interaction with hormones like adrenaline and change the conformation of the cell to allow for communication. This is my favorite diagram. It shows all the different parts. There you have the actual fatty acid tails. You can see proteins that are transverse, or they can also show and kind of sit on top. If they transverse or sit on top, like this one's going to be more for interacting between two different cells or communicating with hormones. There's also ones that transverse. The plasma membrane, because it has that polar region and the nonpolar, acts as a wall or a preventing measure to get in. So the question we have is, how do we actually get things in? Just like when we create a wall, we have to create doors. Our proteins are going to act about like doors, and that's what we'll actually talk about more in class next time. All right, the last thing you need to understand is this term diffusion. Diffusion is the movement of particles from areas of high concentration to areas of low. As you can see, with my diagram over on the right side, there is a lot more particles up here at the top than there is down below, and so the movement of these particles, the randomness caused by chaos, will be in that downward direction. And so that's what we refer to when we talk about diffusion. Things tend towards chaos, and so things randomly move around. If I put a drop of food coloring into a container, the particles are moving around, it will mix it up, and eventually it'll mix it all up completely. This is what we refer to as diffusion. Movement of particles from areas of high concentration to areas of low. Make sure you are able to accurately say that. If I were to spray something smelly in one region of the room, like over here, it would naturally move and spread out evenly throughout the room so that one part doesn't smell any more than the other. Objects fit the shape of their container. And so that's what we really are referring to, random movement. This is a form of passive transport. Passive means does not require energy. Okay, so particles continue to move randomly. That smell, as I used on the last slide, continues to move randomly, but you just can't detect it or see it because it is spelled out throughout the room. All right. There's continuous movement, there's overall change. Once again, things diffuse typically through air, through water, through other liquids. And so smells diffuse through air, food coloring through water. Once again, it's the random movement. There's a bunch of videos. Khan Academy has some. If you don't understand diffusion, not a bad idea to look those up and make sure you understand that by the time you get to class. In this diagram here, I think you should be able to answer these questions. Number one, what will be diffused out of the cell? So what's going to be moving out of the cell? In other words, what color has a higher concentration inside than outside? That'll be moving out. Two, what color will tend to diffuse into the cell? In other words, where is there a higher concentration outside than there is inside the cell? And three, is there a color or an object that will stay the same? This is very common for things that are able to pass through the plasma membrane with ease. So for example, gases. This could represent carbon dioxide, oxygen, and hydrogen. Some will move in, some will move out, and some will kind of be already at equilibrium for the cell. So make sure you're able to answer those three questions. And then once again, this is just an explanation how the role of the diffusion occurs in the plasma membrane itself. Okay, and so we can move particles from one area to another based on principles of diffusion. I want you to write that down. You should have read already in your book about diffusion, so make sure you do that as well. If you have any questions, please bring them to next time. You should have, for the notes on this, what I've kind of asked you to do, bring those to class and be ready to move on to the next section of notes on 7.3. Thanks and have a good day.